Me in the back? Yeah? Okay, let's please get started. I know you're getting lecture exam 2 back in discussion. You got them back today and tomorrow. There's an answer key posted on bSpace with explanations for all 35 questions, as well as the cutoffs for that. If you're kind of curious to know how you're doing, you can look at the cutoffs for lecture exam 1 and lecture exam 2, and then we have the final left. Dr. Patel did not have the full 14 points for iClickers, so we'll make up those remaining 6 points. He had 8 points, so we'll do 6 points on the final for his iClickers that he didn't do, okay? So you're not being... Sure. So the plan was to have 14 points worth of iClicker questions with a maximum of 12, which allowed you to miss some lectures. Dr. Firestone will not have any iClicker questions, so we'll put his 14 points automatically on the final because he will not have any iClicker questions. Dr. Patel was hoping to have 14 questions for up to a maximum of 12, but since he only had 8, that means we're kind of short 6 points. So the way to make up those 6 points is put them on the final. That way each professor gets the same number of points. Is that okay now? So the question was, if you answer those questions wrong, do you get half a point? Let me think about it, okay? Any other questions? Something I have to bring up with the faculty. Yes? So the maximum you can get is 12. So if you had 13 and a half, we would only give you 12. And the reason for that was so that if you came one day and your iClicker didn't work, I didn't have to worry about the 50 students whose iClicker didn't work that day. And or also if you missed one lecture, it wasn't a problem because we had extra. That was the basis of that. Any other questions about that? Okay, so let's get started. So we're going to be doing several phyla today. We're going to be looking at the porifera, the sponges. Not very much time. We have a couple examples in lab. Very brief introduction of that. Cnidarians we'll spend more time on. You'll look at some slides and do a feeding demonstration with cnidarians. We have Platyhelminthes 3 groups. We'll have you look at some slides, look at a live one, and examine feeding behavior. And for the annelids, you'll do an earthworm dissection. And for the mollusks, you will not do any dissection, but we'll have a demo. Okay? So this is the one time where we start to have demos because you just don't have enough time to do the mollusk. We used to have 3 more labs a year, but we don't anymore, okay? So we just try to combine some things this way, okay? So let's just move on. Lab exam 2 is Wednesday night, the 1st. It's going to be 8 to 10 p.m. I mentioned to you from the very start, the reason for that was to allow OChem to have their exam from 6 to 7. There are lectures that you should watch, webcasts, this semester, past semesters. Chem 3D, one of the Chem 3D exams. I wanted to have it then. But I understand that they may not have actually scheduled it then. They may have actually changed their mind at the end. But I can't get earlier rooms. Those rooms are gone. So we're now stuck with 8 to 10. Reviews, there's 4 hours of reviews online. And then, of course, pre-labs worksheets. I occasionally ask a couple questions from the pre-lab worksheet on the lab exam. And there's raised charts. And we have lots of images on our website. Now, I met with a lot of students last week, right before lecture exam 2, basically stating they can't fail, what can they do? And the only response I have is try a different study approach, try this, try that. So I'll go over a little bit again how to study again, okay? Because ultimately the grade you will be given is the grade you earn, okay? Not necessarily what you want, okay? I know that's just the way it is, okay? So charts. People always say, how can I do wellness? Fill out the charts for yourself. We're going to provide you the answers. But the act of going through and making those charts will help you learn a lot, okay? All right? So anyway, here's this ranking, which we did last time, which was the various sections and had the rank. Okay, a lot of people ask me, oh, should I take the class pass, not pass? The deadline was Friday. So I put together this handout, posted on bSpace, send you guys an email about it, okay? But the way this works is, remember, the adjustments will be based, and I brought chalk. The adjustments that will be done in lab are only for the quizzes. Only for the quizzes. And I will know the average for a given section on the quiz, and I know the average for a given section on lab exam 1 plus 2. All students take the same thing. And I said I adjust this up to the easiest. So what this means is no matter what the quiz average is, okay, do you want to discuss this or not? Because I would love to not discuss it. So let's take a vote. Discuss it or not? Okay, because as I said, I don't like doing grades. And so I'd be happy to not do this. Okay, so as I mentioned, I will adjust the quiz scores up so it's on this easiest line. But realize if a section is here to the right, their quiz average will be higher on this graph. It just is the basis of the line, okay? So no matter what the scores are, so for example, and I don't know the averages because a lot of the GSIs haven't entered quiz scores yet, but let's say you're in section 517. They did the best on lab exam 1. We'll see how they do in lab exam 2. I won't know until it's given. But let's say in this section, the average quiz score is only a 1.3. Only a 1.3 out of 5. They still will get a lot of adjustment points because they will be higher on this line than another section. So if a section down here has an average of 3.2, and they're down here, they may be adjusted upward slightly. But overall, this adjusted score here will be higher than that. That's the way the curve works. So if you're in that section that did really well and are really concerned about your average of 2.2, yet you know a friend whose average for their section is 3.6, and they did worse than you, you will be adjusted upward to compensate for that on this fit line, okay? So it's really the fairest way to do it, okay? And then I just said, so basically what you can see here is that, you know, for the section that did highest, there's about a 25% difference between the highest and lowest section. That's true every semester. Anywhere from 20 to 25%. Every semester, there's some GSIs who have the highest and second or third lowest. It's always the case. It's not that the GSIs change, it's the group of students. Likewise, as I mentioned to you before, I can take and look at this group of students in the lecture class. Completely different GSIs, and I find not identical ranking, but fairly similar, okay? So that's why I think it's fair to do it, and that's why I spend the time to do this. For lab exam 2, let me show you the old format, and then you should be thankful that you have the new format, I suspect. I've only heard of one student who last semester did not like this format, the new one. So this is the way the old format worked. You'd rotate from station to station. Station 1 was a rest station, station 2 was here. You had some questions here, and basically here is what you'll see in this week's lab. These are some polyhelminthes and some plexiglass, basically, so you can look to notice some features about that in terms of stream of code. A microscope here that you look through, get in focus, and then an anemone here. And then there's a series of questions. And the timer would be going. And after a minute and 45 seconds, the timer would beep, and then you'd have to go to station 3, the next higher number. And the person who was at 24 went to 25, etc. The person who got to 30, which was the marine tank, would then go to 1. And sometimes you had plenty of time at a station, other times you didn't, but you had to move, okay? So pretty stressful because we thought about putting the timer up, but we had many students say, please don't put the timer up because it would just freak me out. We had many students say, could you please put the timer up because I'd like to know how much time's left. So either way we did it, we couldn't really win, right? So we just didn't ever use the timer. And then this just illustrates another station here, which had the sheet plug that you saw this week, a model of the heart, and a microscope. And then again, there was a series of questions. And you would write your answers on an answer sheet that you carried with you from station to station, and then you got a second rotation of 30 seconds, which isn't very long. It was just enough to have you go through the questions that maybe you didn't quite get done last time, okay? So anyway, and then this just shows you a close-up of this. Okay. The new format, and then this just shows you an example of pinned out earthworm. So what I'm going to do now, and then this is just a microscope slide with a pointer on the structure asking you questions. Now, if you look at the exam reader and you have this, it's going to be a color format. Your exam reader is black and white, but if you go to bSpace, you can print out in color. There's going to be a series of questions that you get to answer during that hour and 40 minutes, okay? There's no rotation, no time. You can spend all your time on one question, or ideally, you'll go through it at a fairly rapid pace, okay? So it's, I think, a lot less stressful. Okay. We won't go to the student who said they didn't like that format. But any questions about the new format? I think it's a lot easier. I did have students from earlier semesters email me to complain that I didn't implement it sooner. And we have this format because we have free classrooms, okay? And we can't do the exams, free exams in a 3-hour period to get them set up, ready for you to go. So that's the basis. So in terms of organ systems, remember this. We went through the organ systems before. I think you're very familiar with them. I mentioned that you should know these cladograms backwards and forward. General comment was from students last semester, even though I went over this repeatedly, how you should know these backward and forward was, I wasn't ready for the cladograms, okay? So you can take that lab exam 2 in the reader and see if you're ready. Please time yourself. You have the answers, grade it, etc., okay? So on the lab exam, I might have a picture of a clam, and then I might have a picture of an anemone, and then I'd ask you what's the smallest group that contains those 2 organisms. And then remember, cladogram works backwards like this, so the smallest group that contains those 2 would be the Eumetazoa, because you got to go backward to a common branch point, okay? So people are saying, I really need to do well in lab exam 2. How can I do well in this? Fill the charts out as you do the lab. Think about, in your mind, what does this dissection look like? What's here, what's there? Can I visualize it? Flashcards. This may be the only time I think flashcards might be helpful, so maybe you can do flashcards. I've taken the diagrams, removed the labels, put them on BSpace, on our website. So you can print out the diagrams without labels. So you can try to diagram unlabeled structures, things like that. You can meet with each other and say, hey, what's the small group that contains this and this? And then the other person has to answer that. Or if you really want to make it challenging, make the various organisms, the name of them, shuffle them, and pick out 2 cards and say, what's the smallest group? Or pick out 4 cards and ask yourself, what's the smallest group? That's just how it will be on the lab exam, so it will give you a chance to think through that cladogram very quickly. Any questions about that? I'm warning you now, again, this is the #1 comment I got last year was the cladograms took a lot longer than what they thought, okay? But they don't have to, all right? And then again, we did this sort of thing. Now we're going to start looking at various groups. So now that as we go through the lab, you can start adding for the cnidarians, the Scyphozoa and Hydrozoa and Anthozoa and that. So you can start adding the small group names, okay? We talked about how tissues are groups of cells that have a common function, and the tissues give rise to organs, and then organs within organ systems, okay? And we talked about diffusion. This formula here is critical. Evolution can affect the K, that's the constant of the chemical or the molecule. Cross-section can be modified during evolution. Change in concentration gradients, potentially, the organisms can potentially make a concentration gradient. And then the distance that something has to diffuse can be altered. The second lab, when you look at those Nitella cells, they're huge. And the time for a molecule to diffuse from one end of the cell to the next would be way too long to support life. So the way they overcame this limited diffusion was bulk mixing of the fluid. You had that cytoplasmic streaming. Okay? So that was one way it evolved a mechanism to overcome those rates of diffusion. And just remember, it is a rate, and it's cross-sectional area, the length, and the concentration difference. Okay? We're going to see this sort of in, for example, things that are flat or small, the area diffused is sufficiently short enough, or surface area is large enough, that you don't have specific circulatory systems. So for example, in protozoans, the amoeba, it's a thin layer there, diffusion is fast enough for gases, etc. In terms of the Hydra here, it's pretty much 2 cell layers. There's the outside layer, sometimes called the ectoderm, and the inside layer, sometimes called the gastroderm, because this is where feeding is going to take place. It's 2 layers. So gases are free to diffuse from the outside into here, or from this gastrovascular cavity into the cells. Once the gastrovascular cavity gets closed up, though, realize that you may be depleting the O2 concentration or increasing the CO2 concentration in here, so then you might be more dependent upon diffusion from the ectoderm. Okay? And this is where feeding will take place. We'll go over that, okay? And you can see there's this opening labeled mouth here in the Hydra. It would be better labeled mouth/anus, because your solid waste have to be released from that opening as well. All right? And we said that basically we're tubes within tubes. So we have the outside body wall, we have the inner GI tract, which is the other tube, and then we have that packing of cells, etc. inside there. And remember, we have cavities. So we'll see the circulatory system interplays with the respiratory system for gas exchange at the lungs, large surface area. We have interchange at the kidneys in terms of nitrogenous waste, ionic balance. We have to have gametes get released, etc. So anyway, here's that GI tract, mouth, anus, specialization along here. You saw this in the rat, stomach for storage. The vast majority of digestion and absorption occurs in the small intestine. All right? We went over the coelom. Remember, we're talking about a true coelomate organism that has the GI tract with peritoneum around it, including this coelom. And we will be looking at the platyhelminthes. They are in the protostomes. They do not have a true coelom, the solid packing of cells. Okay? So let's go over a few things here. Here's that gastrovascular cavity. Again, that mouth/anus. Here shows you the 2 layers. Here they call it epidermis. It could also be called ectoderm on the outside there. Here's that gastoderm for feeding. Pretty much one cell layer, and there is some gelatinous layer in between here, extracellular matrix. What kind of cells typically would be producing extracellular matrix like in a human? Epithelial, nervous, muscle, or connective? Connective tissue. In this case, they pretty much have just 2 layers of epithelial alone. In terms of mating, we have both asexual and sexual reproduction. We'll talk about both. Remember, sexual reproduction, formation of gametes, generation of diversity, fusion to generate even more diversity, okay? And then development, we have an entire lab on this, but what we'll talk about is the formation of the zygote. When 2 gametes fuse, we'll form the zygote. So the ploidy of this is 2N, 2C. We'll go through a rapid series of mitotic divisions. And I don't know if you can tell this very well, but this is the same size as that. That's pretty unusual. Usually when cells undergo mitosis, they get bigger, so they're about almost twice in volume, and then they go through that mitotic division and cleave and get 2 daughter cells. Cleavage is unusual. You get rapid cell division, and these cells just get smaller and smaller and smaller. That's pretty unusual. But by getting many cells, we can form a cavity within here called the blastocyst, which will allow cells to migrate inward to form the germ layers. And we'll talk about 3 germ layers versus 2, etc. Okay? So we'll do that a lot more in the reproduction development lab. This just illustrates those cells migrate from the outside surface inward. And in order to move, they have to alter their connections with one another. They have to become motile. And as they do this, clearly they're interacting with new neighbors. And as they interact with new neighbors, they get new gene differentiation, things like that. So then this can become one layer called the gastroderm, and another layer, the mesoderm, and the outside one, the ectoderm, in terms of germ layers. So we'll talk about 3 germ layers eventually. And they are the endoderm, they are the mesoderm, and then ectoderm. Okay? And we'll do that again. All right, so we'll talk about protostomes versus deuterostomes. As you know, in a protostome, the first opening is the mouth, the basis of proto, first stome mouth. And in deuterostome, the other opening, second opening is the mouth. And these are broad generalizations. Broad generalizations. That doesn't mean that every deuterostome does this exact pathway or every protostome does this exact pathway. These are broad generalizations, okay? Different organisms have adapted different mechanisms during evolution. This was a problem last semester because the professor in the 1A class gave a specific example where a deuterostome did something different. He did an example class and then asked an eye clicker question on it, and the students didn't look at the specific example, but with the generalization. So remember, generalizations, broad generalizations. Here's these cladograms again. You should be pretty familiar with these. I think the one in the lab manual will be most useful for you. This just sort of emphasizes that when we get to the protostomes, we have 2 large groupings, the Lophotrochozoans and the Ecdysozoans, okay? And for the deuterostomes, we've already looked at chordates, so the remaining group we will look at will be the echinoderms. Okay? And we'll do that next week, the echinoderms. In terms of the Lophotrochozoans, we will look at the platyhelminthes this week. We'll look at the annelids this week, and we'll look at the mollusks. So this kind of focuses on the Lophotrochozoans this week, okay? And then in terms of the ecdysozoans, these are the ones that shed. The two groups, the one group we'll look at for sure will be the arthropods. And you've had a little bit of information about nematodes from Dr. Patel, and I might talk briefly about them and maybe arthrophorans if we have time. Okay? I certainly want to show you a video of these. So at the end of the lecture, I'm going to try to end about 5 to 10 minutes early, and that's to show you some images, okay, some movies, if there's time. This is showing you a sponge. Basically what they've done here is they've added some dye, so you can see that this dye is coming in through small pores, and then all of it goes out through this opening there, okay? And then this illustrates for you that opening is called the osculum. Now, on the lab exam, I could potentially have a diagram of a sponge and ask you to identify a couple cell types. I probably wouldn't, but I don't want to. I mean, I can't. But when you study for the lab exam, look at the big picture first, identification, big things. Then if you want to start focusing on the really small details, do that at the end. Prioritize your studying, okay? So I probably wouldn't ask you, is this the osculum or not? I could, but I wouldn't most likely do that anyway. So a couple cell types that I do want to talk about. One is the choanocytes. Basically, that means collared cells. What we see here is the cell here, site, choanocyte. It has this collar here. It has a flagellum here. What this flagellum does is as it beats, it basically creates a current. And what it's going to do is it's going to create a current that forces the water out this osculum, and therefore it creates basically a negative pressure, and then water enters into these pores. Okay? And as they enter through these pores, the water passes through these collar cells here, and then food gets filtered out. So they're filter feeders, basically. And they can discriminate between food and non-food. The food gets taken up by phagocytosis, so the digestion here is intracellular. Intracellular because it's occurring within the cell. With humans, we have extracellular digestion. We dump digestive enzymes into the GI tract. Okay? The advantage of that, we can eat really large food, right? We just have to fragment it, and then clearly by the time it gets to the GI tract, we have to fragment it small enough, increase that surface area for breakdown and absorption. And then there's a second cell type, the amoebocyte. So these things lack true tissues. Okay? We get tissues with cnidarians, but these really don't have true tissues. So if you look at the Cladogram, the name for animals is metazoans. And we have a branch point here where we have the sponges, porifera. So this is parazoa, because they lack true tissues, and these are the eumetazoa. They have true tissues. Okay? Then we'll be doing all our branch points from this branch of the Cladogram. We're through with this branch point, okay? Any questions about the sponges? The amoebocytes help to distribute some of the nutrients, and gametes are just basically made by these two types. There's no gonads. Question? Yeah. So what happens is basically there's a current created, and as this water gets moved in, hopefully there's food suspended in it, and as it passes through these collar cells, basically there's a net here with some mucus-like surrounding part of it, which captures it and then gets taken in by phagocytosis. And then these amoebocytes can actually crawl through here and help distribute those nutrients. So they don't have a circulatory system to distribute nutrients. The amoebocytes help to do that. Okay? Any other questions? Great. Let's move on. In terms of cnidarians, the 4 classes we'll talk about: the hydrozoa, which are the hydras. So if you could cross out this Portuguese man of war, that's not really a hydrozoan. It's a kind of bizarre organism that's used a lot to talk about this, but it's 4 or 5 different groups of organisms that come together to form this one organism. So let's forget about the Portuguese man of war. We have hydrozoa, we have scyphozoa, the jellyfishes in that. We have cubozoa, these are the box jellies. They look like a box almost. They actually, I'll show you an image of this. They have eyes on the periphery, fairly complex eyes, but we don't think they form images, but I'll go back in a little bit. And anthozoa, which are the anemones and corals. And you've heard a lot about corals dying off. This is one documented case of transmission of a human causing disease in another organism, and that is there are some corals that are dying off of Florida, and basically it's from a bacteria and human waste, feces, sewage, things like that, and it's now infecting the colonies of coral and killing them off. So 2 body forms that we'll talk about here. There is the medusa form, which pretty much only, the jellyfish pretty much only have the medusa form. Looks like this. And then they can, in this case, these are the adults, make egg and sperm, fertilize zygote. And then there's a polyp form, which is stationary. And we'll see that in the hydrozoa. So the hydrozoa that we're looking at only has the polyp form, but some of these cnidarians have both forms in their life cycle. Okay? But the hydras that we're looking at are freshwater, and they only have the polyp form. Here's the chart that I was talking about. I think it's not really useful for me to just read off this chart. I've always hated that when an instructor does that. But the thing I do want to point out, a couple key things. Number of germ layers. They only have 2 germ layers. Okay? They do have tissues because they have common functionality, but they don't have true organs because they only have 2 germ layers. Okay? So they have ectoderm, and they have endoderm, but no mesoderm. Okay? During development. So this would be called diploblastic is the scientific term. I'm going to write it over here. Diploblastic. So we have the platyhelminthes off here. Sorry, the cnidarians. And then we'll have the triploblastic here, which means they have 3 germ layers. So the one that's missing here is mesoderm. And then they do have ecto and endo during development. And then the triplo, of course, is all 3. Okay? So on this chart, metamerism, stats refers to repetition. I mentioned it last week, but it can be a little confusing. It's repetition of organs, whether they're internal or external. For the arthropods, we'll see they have a lot of appendages.